Hello and welcome everyone to our next lecture. In this lecture we're going to talk about transcription. It's this first step, well really second step in this overall process that we've been talking about how DNA makes RNA, which makes proteins. And so we're going to talk today about how we go from DNA to RNA. The last lecture we talked about how DNA goes to DNA, which is DNA replication. Now we're going to talk about DNA transcription. So remember, this is just getting back to this, what is called central dogma of molecular biology. And in this term, I just want to point out that sometimes you'll hear dogma as a term used to describe an incorrect prevailing thought. In this case, this is a very much correct prevailing thought. It's basically, you could say, the central idea of molecular biology, that DNA encodes for RNA, which encodes for protein, which in turn make up what you are as an organism. Okay, so we've already talked about how DNA can just continue to copy itself, but let's talk about this step, DNA to RNA. So up to this point, we've talked about the structure of DNA, and we've briefly talked about DNA replication. Now let's talk about that DNA being transcribed to RNA. So transcription. If I ask you to transcribe something out of a textbook, I'm asking you to write it down in the language that that textbook is written in. Right? You would write it down in the same language. That's the same idea when DNA is transcribed to RNA. We go from a language of nucleotides to the language of nucleotides. Remember what a nucleotide is from last lecture. When we talk about the nucleotides, there are 5 nucleotides in DNA and RNA. Remember that adenine and guanine, A and G, are what we call the purines. And they're purines, and they're the same structure. They've got 2 of these nitrogenous rings. And then T, C, and U, thymine, uracil, and cytosine, are what we call the pyrimidines. And remember, I gave you that little mnemonic. TCU is a top of the pyramid school. And that's how you can remember T, C, and U being the pyrimidines. And they only have 1 nitrogenous ring. Okay, so let's get into it. The transcribing of DNA to messenger RNA. Now we have to put M before RNA because there are other types of RNA. But when you see mRNA, that is a messenger, and the message that it is carrying is one of how we are going to build these proteins. So the transcribing of DNA to mRNA is relatively straightforward. 1 nucleotide being added to the mRNA strand for every complementary nucleotide that's read in the DNA strand. It's very, very simple. Transcription is the same in prokaryotes as it is in eukaryotes, except for one thing. It occurs in the nucleus of eukaryotes. Where would it occur in prokaryotes? Well, just in the cytosol, because there are no nuclei in prokaryotic organisms, right? The nucleoid region is just the area in which genetic information is held that doesn't have a membrane around it. Whereas in eukaryotes, the DNA is sectioned off in its own membrane called the nucleus. In both prokaryotes and eukaryotes, transcription occurs in 3 main stages: initiation, elongation, and termination. We'll talk about all 3 of those right now. So let's get into it with initiation. Transcription requires the DNA, remember, double helix. DNA is a double helix while RNA is a single helix. It requires that DNA double helix to partially unwind in the region in which the mRNA is going to be synthesized. Just as DNA replication needs the DNA double helix to unwind. They need the enzymes that are going to do this need access to those nucleotides. That region of unwinding is called the transcription bubble, whereas before when we talked about replication, it was called the replication bubble. So it's just a little bubble that forms in the DNA double helix so that these enzymes can get at these nucleotides. The DNA sequence onto which the proteins and enzymes involved in transcription bind to initiate the process is called a promoter. In most cases, promoters exist upstream of the genes that they regulate. So that just means that if you were, let's say that you were on a boat and you were traveling down this gene, the promoter would be upstream and downstream would be the genes in which they control. The promoter allows for some genes to be transcribed more than others. And why would some genes need to be transcribed more than others? Well, think of it this way. If you have a gene, let's talk about E. coli, okay? E. coli are a bacteria, and they have the ability to break down sugars for their energy. Now, especially they like, milk sugars, okay, lactose. Now, lactose is a really good energy source for every organism that can break it down. But think about where E. coli are found. They're often found in the gut of organisms and obviously in fecal matter. And there's not that many times in which they will encounter lactose in their environments. So in order to break down lactose, you have to transcribe these genes that will then make the proteins that can produce the function of being able to break down this lactose. If you are not encountering lactose in your environment, why would you want to spend the energy that it takes in order to transcribe those genes? And so basically, these promoters are able to shut off the transcription of those genes until they do happen to encounter lactose in their environment, and then the promoters promote the transcription of those genes. And so that's basically all I want to get into it. There's a whole process involved with that, but that's a discussion for another day. But just take away from it that promoters regulate how often a gene is transcribed, because to transcribe genes takes energy, and you don't want to transcribe genes when you don't have a use for what they're producing. So here's a look at basically what this would look like. Here's the promoter, and your RNA polymerase enzyme would attach here when it's ready to start creating this messenger RNA. And remember that there's a direction in which this thing is synthesized, meaning that the nucleotide can only be added on to that free 3' end. If you remember that, the free 3' end, it goes 5' to 3', and you must add nucleotides onto the free 3' end. So that would be the direction of synthesis, adding this way. And basically what happens is the mRNA starts to form this link of nucleotides, and it's eventually will pop off, but we'll get to that in a second. Notice that there is a template strand and then the non-template strand. So only one strand of the DNA gets read at any one time. So the next step is elongation. Now, transcription always proceeds from one of the two DNA strands, which is called the template strand, like I said. The mRNA product is complementary to the template strand, and in that sense, it will be almost identical to the other DNA strand, which we call the non-template strand, but the only change would be that RNA, of course, contains uracil in place of thymine. So on the template strand, wherever there was an A, on the non-template strand, there would be a T. But since we're producing an RNA, a messenger RNA, wherever there's an A on the template strand, there will be a U in the messenger RNA. Okay, so that's important to keep in mind when I ask you to transcribe mRNAs from a template strand of DNA, which I will do on the exam. So here's what this process would look like. A little more in depth. Here's the same RNA polymerase enzyme, and you can see here's our DNA template strand. We're adding on to the 3' end. Here's the direction of synthesis, adding on to the 3' end, not the 5' end. The 5' end is where you can't add on to nucleotides, okay? Remember, look at how we're reading in the 3' to 5' direction, because these molecules are anti-parallel. Okay, so if you read the 3 to 5, you produce a 5 to 3. Okay, and it's it could easily be the other way. If I put the template strand as 5 to 3, I would ask you to write the new mRNA in the 3 to 5. Okay, so that's important to keep in mind, is that the anti-parallel nature of these molecules. So take a look. This is TACGG. Okay, that's the start of that template strand. And on the non-template strand, we have, of course, ATGCC. Now, on the mRNA, we should have the same thing, except wherever that T is, there should be a U, and we do. We find a U instead of T, G, C, C, and so on and so forth. And so that's basically, you're all pretty familiar with this by now, I think, where the A's and the T's go together in DNA, the A's and the U's go together in RNA, and G's and C's go together all the time. So that's elongation, where we're just continuing on the synthesis of our RNA. At some point, we've got to stop, though. We've got to stop because we've made our messenger RNA. We know what we need to create in terms of proteins, and that is termination. So once the gene is transcribed, the polymerase needs to be told to stop making an RNA and dissociate, get off of the DNA template strand, and also release or liberate the newly made mRNA. In prokaryotes, the newly formed mRNA would actually have already started being used to make proteins. And the reason for that is it's all happening in one place, the cytosol. So eventually what you'll see is that ribosomes will read the mRNA and link amino acids together to make proteins. And so in prokaryotes, that is happening as the mRNA is being produced. It starts to tail off of the RNA polymerase enzyme, and they attach onto it. Well, I'll show you that in a second. But before we can do that in eukaryotes, the mRNA has to undergo some further processing because it's got to be transported out of the nucleus and into the cytosol. Here's what that looks like in prokaryotes. So here's our DNA in green, and you can see, you don't see it here, but somewhere along here there are enzymes called RNA polymerase, and they're making the mRNA, which is that blue line. And here are our ribosomes in red already attached to the mRNA, and if there's many ribosomes on the same mRNA, we call them a polyribosome. And what they're going to be doing is just making the proteins. That this blue mRNA codes for proteins, and they're already making the proteins before this thing is even done transcribing. And so that's very nice, and it obviously happens very quickly. But in eukaryotes, that doesn't happen that way. So we have to do some processing of the RNA. The newly transcribed eukaryotic mRNAs have to undergo several processing steps before they can be transferred from the nucleus to the cytoplasm and then translated into a protein. Now, the reason that we have to do these processing steps is because sending out molecules into the cell, you've got to put special markers on molecules that say, hey, don't break this down right away because it's something special and something important that we need. And because cells can't talk to each other, they've got to do that through processing and putting on special markers on these molecules that are, in fact, their language. The first processing that happens will create a molecule that is much more stable and will last longer than a prokaryotic mRNA. And it's got to last longer because it's got to move through the cell. Once elongation is complete, and an enzyme, you'll take an enzyme and it adds a string of approximately 200 adenine residues to the 3' end. So basically on the tail end of this mRNA, you add a bunch of A's. And we call that a poly A tail. So it would be A, G, C, A, U, 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 whatever. And then finally A for 200 A's. That doesn't code for anything. It's just something called a poly A tail. And that modification just further protects this pre-mRNA from degradation, and it signals to other cell factors that this transcript needs to be exported to the cytoplasm. Don't break it down. Get it out to the cytoplasm. It has a job to do. Now, the next thing are these introns and exons. Eukaryotic genes, you've got in your DNA billions of letters, A's, T's, G's, and C's. But some of them are going to be protein coding sequences, which we call exon. They're called exon to signify that they are going to be expressed. And then there's intervening sequences called introns, and the intron denotes their intervening role. So some sequences in your DNA just don't code for any proteins. They're just there, and they don't code for proteins, whereas some of the sequences do. Introns get removed from the pre-mRNA during processing. Intron sequences in mRNA do not encode for functional proteins. That's why they're intervening. They are not expressed like the exons. Okay, and the reason that genome, in your genes, have these introns and exons is it gives your DNA a way to make many different proteins from your DNA. And I don't want to get too much further into it in this class, but just understanding that exons are expressed and introns are the intervening sequences, and the introns get removed during processing is enough. If this is messed up, though, by even a single nucleotide, major problems will occur. If you take out an A or a T and a G and a C in the wrong space, you are suddenly making a very different protein from which you should be making, or perhaps not making a protein at all. And so obviously it's very important to get these sequences correct. The process of removing introns and then reconnecting exons is called splicing. So here's what this looks like. So here's that primary RNA transcript right as it's made, and you can see there's exon 1, 2, and 3. You've got to put those together, and to do that, you've got to splice out these introns. And so that intron, those introns get spliced, and also this poly A tail gets added, which means, hey, don't break this down. We need it. We need to get it out into the cytoplasm, into the cytosol, out of the nucleus. And here's another piece of RNA processing called the 5' cap. Not as important to worry about. It's just another example in which this is saying, hey, don't get rid of this. We need it. Okay. So anyway, all I want you to remember is that we splice out the introns, connect the exons, and we add a poly A tail so that we can keep this thing around and it doesn't get degraded right away. Okay, so that's basically what transcription is all about. The next step would be translation, where we take our fully processed mRNA out into the cytoplasm, into the cytosol, and the ribosomes get to work on it. So that'll be the next lecture, but let's work on some exam questions here. So if I gave you a template strand and I said transcribe this DNA, how would you go about it? So first of all, the directions tell you right off the bat, this is DNA. The other way, if it didn't say for some reason, if it's DNA, how else would you know? Hopefully you understand that you would see T's and know that this is a strand of DNA. The second thing that I want you to take note of is I have now added 5' and 3'. So I'm giving you the direction of this molecule, 5' to 3'. And so if you're going to keep the anti-parallel nature of this molecule intact, your answer has to have the correct placement of the 5' and 3'. So hopefully what you're remembering is that if this goes 5' to 3' in this way, your answer has to go 3' to 5' that way because it's anti-parallel. Okay, the next thing is I tell you to transcribe it. So what are we doing here exactly? We're going from nucleotides to nucleotides. And we are making a transcription, which means going from DNA to RNA. So basically what you should come away with is, well, whenever I see an A, instead of a T, I'm going to put a U because we're now making an RNA, and then G's and C's go together. So hopefully you should make something like this, where you've got 3' to 5' because it's anti-parallel. And sorry, I'm actually even giving you a further step. This is our non-template strand of DNA. Here's 3' to 5'. Now, what you could take from right there is literally, if I gave you that, all you'd have to do is copy that and then replace the T's with U's, and you've got your answer. That's your transcribed mRNA. If I don't give you the non-template strand, it's the same idea, just making sure you're going 3' to 5' and releasing, changing the T's with U's, okay? And so that's how you would transcribe DNA. Here's another example of maybe a multiple choice question. The following DNA strand is used as a template for transcription. Okay, so we know it's DNA, but we also can see the T here, and we are going to use it as a template for transcription. So which of the following RNA strands will be produced? Okay, right off the bat, first thing I would do is note 3' to 5'. So my answer has to be 5' to 3'. Then I look at my answers and I notice, okay, all of them are in that order already, so we're good there. The next, I would just figure out what sequence it is. And I would read it left to right, C, G, T, A, A, but let's just go with the first letter. C, so what would the 5' to 3' RNA transcription need to be? Well, the first letter would have to be 5' G. Do we have that? Well, here's 5' G. The next is 5' C, 5' A, 5' U. Well, none of the other answers have that, and so it's got to be A. Should be a really easy question here. 5' G, let's just make sure. G to C, T to A to A, A, U, U. So we can see that this works. That's our answer. The answer is A. Now, it could be trickier than that. What if I put 5' GCA, the correct answer, but right below it I put 3' GCA UUC. Okay, I put the exact same letters, but I switched 3' and 5'. Well, if you select 3' to 5', when the template is 3' to 5', that's incorrect because that's not anti-parallel. And so I hope that makes sense for you guys. Of course, always give me a shout via email or message if you don't understand. And then when we get to our next lecture, guys, we will talk about translation. The next step in this process from going from DNA to RNA to protein is, of course, translation. And I said when you translate something, you go from one language to another, and that's what happens in this process as well. We go from the language of nucleotides to a language of amino acids. And so that'll be our next lecture, and then we'll get ready for what comes after that, the exam. And so hope this one made sense for you guys. Let me know if it didn't, and I'll see you guys in the next lecture on translation.